Yeah, so it is my pleasure to introduce today Dr. Sarah Dyer. Sarah is the non-vertebrate genomics team leader at EMBL-EBI in the UK. She leads Ensembl plants, metazoa, and outreach teams, and is responsible for the UK components of Wormbase and the Alliance of Genome Resources. Prior to EBI, Sarah was a group leader at NIAB, working on crop diversity in cassava and common bean, collaborating with CGIAR gene banks. Her background is in bioinformatics and crop genomics. And Sarah has agreed to join us today to talk about Ensembl and particularly Ensembl plants. And so, yeah, take it away, Sarah.
Great. Thanks, Peter. Thanks, Anna-Rita. Thank you for the opportunity to be here. Let me share my screen. How's that looking? Okay, I can't see anyone, so I'll
Great,
take the
great.
silence that it's working.
It's fine.
Lovely, thank you. Okay, great. So yeah, my topic is going to cover kind of plants, pollinators, and pests, and give you a bit of a tour of Ensembl. So I'm based at EMBL-EBI in the UK. So stands for the European Molecular Biology Laboratory. So we have 6 sites. Our headquarters are actually in Heidelberg in Germany. We're the second largest site up here, EMBL-EBI in the UK. So we're just south of Cambridge. And then we also have sites in Grenoble and Barcelona, Hamburg and Rome. So the kind of mission of Ensembl is around, there's a lot around delivering data resources and services. We also have some research component. We do a lot of training for the next generation, there's industry engagement, and then we have a kind of role to play in coordinating bioinformatics in Europe as well as part of our kind of intergovernmental status. In terms of the data resources, this is certainly one of the things that we're known for. So we have kind of a few different resources listed here that I'm sure you'll be familiar with, some of them ranging from our kind of chemicals and molecules kind of going along the spectrum. We have genes and genomes, which is where Ensembl falls, also the ENA, which is where all our sequence reads get kind of deposited, an expression atlas, which has kind of is the home of expression data at EBI. And then we also kind of work very closely with the groups, the different protein resources. So we have the AlphaFold database, PDB Europe is there, and Uniprot is based with us as well. So we have lots of connection into those groups. And then there are other groups as well, kind of the Bioimage Archive is worth a mention. That's a new one for kind of imaging data. So it's relatively new compared to the rest of our services. And then we have the European Variation Archive, which I think you might have had a webinar on not so long ago. And then yeah, we also connect through to Europe PMC and the literature side of things. So we kind of span all of these different services. So in terms of what Ensembl is, if in case you're not familiar, it's an open access platform for genomic data and annotations. If you come to ensembl.org, it looks like this as your kind of landing page. So we host genomes and gene annotation data, genetic variation, and we run a lot of comparative genomics analyses. We provide a web browser, data available for kind of downloads. So I know lots of people will come and kind of just take data from the FTP site after a release. And we also have APIs to interact with the data. And we develop some tools, and the variant effect predictor is one of our most popular, which I will come back to later on. And everything is completely open source. So we have all our code in GitHub and all the data is on the FTP site. So in terms of how this kind of meshes with AgBio data, some of the key working groups that I've been involved in, and also my colleague Peter Harrison, who was at EBI before and was a kind of a key player here in AgBio data, these are some of the ones that sort of were very key for us. So I've been part of the genome assembly and annotation working group because obviously we host a lot of genomes, and having a well-named genome assembly is a sensible thing so that everybody knows what they're talking about. We're also very keen on the standards for genetic variation through our kind of links with EVA and the data that we bring in there. And pangenomes are a kind of key sort of new adventure for us. So we also are quite kind of we're happy to see that group and the recommendations coming from there, which have been quite useful for us already. So in terms of what we cover, In the early days of Ensembl, we were a vertebrates only show. So it was kind of centered around human and mouse. This is a snapshot of today, the kind of different species diversity that we have. So we still have quite a lot of vertebrates over here, but we have an awful lot of metazoan species, so a lot of invertebrates, a lot of fungi, and plants and protists as well. So we have quite a lot of diversity going on. Um, and we have different component sites of Ensembl that capture those different areas of the taxonomy. So if you go to ensembl.org, you'll only get to the vertebrates. But if you're interested in plants, you go to Ensembl plants, metazoa to Ensembl metazoa, and so on. And we also have, just because my taxonomy was only eukaryotes, but we also have Ensembl bacteria, which has around 30,000 strains kind of banked in there as well. And in terms of what are our kind of most popular species, we tend to have the sort of model genomes that we've brought in are very popular, but there's an awful kind of lot of agriculturally relevant species that people want to use. So for plants, this is our kind of top 10 most accessed species. So we have wheat, we have Arabidopsis, of course, rice and maize and barley, tomato, grape, soybean, potato, and rapeseed are our most popular. In the metazoan space, we have the models, kind of Drosophila and C. elegans. We have some mosquitoes, because vectors of disease is another kind of area that we connect with. And then we've got kind of honeybee, silkworm, red flower beetle, and then some more marine. We've got kind of oyster and these couple of models over here, the sea anemone and sea urchin. In the fungi, we have models, so we have kind of the yeast kind of Cerevisiae and Pombi, and we've got Aspergillus, but then we also, a lot of the kind of different fungal species are actually linked to disease, so kind of Magnaportha, Botrytis, Fusarium, Septoria, species like that. So in case you can't recognize them from their thumbnails. And then I went through and pulled out some other kind of ag data species based on the databases that are ag members. So in livestock, whilst I've only got cow represented here, we have through Peter Harrison's connections, we have quite a lot of other livestock species. So we have quite a few pigs, and we have chickens and horses and sheep species, sheep strain breeds as well. And then we have other kind of gone more into the plants. So we've got cassava, we've got golden delicious as our apple representative. We have a few prunus, so we have kind of cherry and almond and peach, rose. Peanuts is a recent addition for us. Pigeon pea, lupin, common bean, peas, red clover, a few Vigna species. So we've got the adzuki, the mung bean, and the cowpea, banana and yams, and others that too many to fit on the slide. But these are some of the species that I wanted to call out given the audience. Um, an activity that we did relatively recently was bringing in some pest genomes into ensembles. So these were coming from the Pest Genome Initiative, which was published in 2023 and was a collaboration between Bayer, Syngenta, and Rothamsted Research to produce genomes and annotate them. And then through the EMBL-EBI industry program, we connected with Bayer and Syngenta, and they asked us that we would bring the pest genomes in. So we loaded 24 of them and added kind of missing annotation onto half a dozen that, for whatever reason, didn't have annotation available. And so we have a blog post about that. You can have a look at our blog. But these are just some of the species that we brought in to kind of improve our representation of pest genomes. And so in terms of where the data comes from that we host, so we have one kind of strict rule that all of the genome assemblies that we bring in must be available from the INSDC, so the International Nucleotide Sequencing Data Consortium. So this is the kind of the big three of the DDBJ in Japan, ENA based at EMBL-EBI, and NCBI. So those kind of sequence archives. So for us to work with an assembly, it has to have been deposited, and it has to have a GCA identifier. Otherwise, we're not allowed to work with it because we have an agreement that we won't work with it otherwise. So for us, that's sometimes tricky because people come to us asking for us to import a genome, and we're actually not able to unless it's been deposited. So that's something that we always have to kind of navigate. Beyond the assemblies, we're more free. So in terms of the annotations, sometimes we annotate things ourselves. So our Ensembl gene build team, traditionally in the vertebrate field, they produced all their own annotations. They're now expanding across the tree of life. So we have a number of annotations coming from from within our teams. But traditionally, we've brought in for outside of vertebrates, we've always brought in community generated annotation. And I use community a little loosely because it can be coming from a community group or it could be coming from RefSeq. So just kind of external annotation is what we would bring in, mainly for like plants and metazoan, for instance. For the genetic variation data, this is of course coming from the community. It can either be passed to us directly, but we much prefer it if it goes into the European Variation Archive first, and then there's a record that we can kind of link out to. So that's then there for everybody to kind of see and reference, and there's benefits to depositing your data there. So we kind of encourage that. And then with gene expression data, we currently pull from the Gene Expression Atlas. So again, that requires kind of data deposition, and they process it, and then we just kind of mirror what they have. So that's where our data kind of primarily comes from. And then we also link out to other resources like Uniprot and kind of connect to some of the other key resources that are useful for people. In terms of the sort of priority areas and the species and how we kind of go about bringing this data, sometimes it will be based on specific collaborations. So if we're part of a group, we're part of a consortium, then we'll bring in the data related to that kind of project. Other times it's through user requests. So we have a very active help desk and people get in touch and ask us for species, and we do our best to bring them in. And then we have our own kind of priority areas that have sort of emerged over the years. So human remains our most important species and will continue to do so. Model species are also, our users really like to use those to bring them in. I think they're a good way for people to connect from starting from an annotation and move out to kind of less annotated species. They kind of provide that way in. So we kind of continue to import these model species data. Disease vectors, I mentioned before, has been an area for us, but we're increasingly seeing, particularly in the plant sphere, so much of the species that we have some kind of agricultural relevance. And we're also involved in lots of biodiversity projects, which I will say a little bit more about. But in a way, those are able to bring in other species, which whilst they may not be the target of kind of agriculture, they are relevant to the ecosystems and to the environment. So we're also picking up other agriculturally relevant species coming in through the biodiversity projects. So you can see that we're involved in many biodiversity projects from this slide. So lots of them come under the umbrella of the Earth Biogenome Project. Peter Harrison's work was kind of connecting with ERGA for the reference genomes and the vertebrate genome projects, and then FANG and Aquafang and these kind of projects around the sort of livestock and different annotation of animal genomes. The human pangenome is another important one for us, that we're part of the Human Pangenome Reference Consortium. But the one that's giving us the most kind of biodiversity data sort of immediately is the Darwin Tree of Life project. And that's a collaboration where the assemblies are generated and come to us from the Sanger Institute, and we provide the annotation coming from Ensembl. And so this, if you're not familiar, this project aims to sequence 70,000 eukaryotic species from the UK and Ireland. So it's quite an ambitious project, and there are already several thousand genomes in. So yeah, that's a source of a lot of our data right now. And when we get data in, we have a release cycle. It has to be processed before we can put it out. So we'll bring in assemblies, we'll bring in gene sets or generate them ourselves. The longest part of our release cycle is the comparative genomics that we do. That really takes some time. We'll bring in variation data and then put everything out. In terms of our kind of releases, so we've got here that it takes 3 months. It's kind of 3 or 4 months between releases, but really the releases are overlapping, so they actually take us a little bit longer than that to run in-house. And we're on our 60th release of Ensembl genomes and 113 of our vertebrates that kind of was the original. But that's like a really long time. If you're waiting for your data, that could be quite long that you're waiting for. And with all of the genomes coming, particularly through the biodiversity projects, we really needed to sort of move things more quickly. So a few years ago, we invested in something which is called the sort of developing the rapid release system. So it's a very kind of lightweight initial processing where we bring in the genomes and the annotation, but instead of doing like full comparative runs and regulatory builds and some of these things, we kind of just put out some simple homology predictions instead and get it out there, and then we have the opportunity to update later on. So this is then we're having releases every couple of weeks rather than quarterly as we kind of had been doing. So we've just announced that we've actually frozen our rapid release cycle. And so all the data that's in rapid release, it will stay there, but we have also lifted it all across to our new beta site. And this is a screenshot of our beta site, and I'll give you a bit of a tour of that at the end. So if you go to our current site, I'm going to use plants as an example, but they all look the same, all of our current sites for now. So you have a kind of landing page, you can explore species, you can just jump in straight with searches. You can get back to our archives as well. If you get into the genome browser space, we have these kind of different levels to orient you. So we'll start off by showing you, if you're at a given location where you are on the chromosome, a much more zoomed in view of the kind of the local area. And then this is focusing on our gene of interest. And if I just show you the bottom part of that screen, again, we've got the gene of interest. And we'll also show things like the genetic variants here, which are color coded by their kind of expected severity and impact on the protein. So all of our views have these little kind of cogs in the corner, and you can configure a lot. You can switch tracks off and on and change how they look like. There's a lot of functionality there as well. And we tend to have things arranged by, you could either be searching by a location or maybe by a gene, and then the gene will have transcripts. And each of them will have specific menus with different things that you can see and do. So one of the things just to kind of draw your attention to here, we're in the transcript view. So we have, you can export the sequence, you can learn about the protein. And if we've got an AlphaFold predicted model, then you can click on that and it will take you to the kind of 3D structures as predicted by the AlphaFold kind of approach. And so typically we'll show this here. This is all interactive. I didn't dare the live demo. But we have model confidence kind of showing those regions which are very certain, folded with very high confidence in dark, and then the kind of more or less confident regions that tend to be less structured, sort of orangey scale. But the nice thing that we can do is we also have a number of menus here that you can switch on other features and show them on the structure as well. So if you're interested in a particular variant and you want to see where it is in that 3D structure, you can switch that on. You can highlight and hover and it will kind of highlight one, or here I've just turned on all the deleterious ones that are showing up as little kind of red bits. So you can have a look around and see which of those variants are likely to be impacting the kind of part of the protein you're most interested in. In terms of I suppose up until now, or until recently, we've been at a point where we've only hosted one genome for a species. We've had the kind of reference genome per species has been our approach. And now that we're in pangenome times, we're expanding that for certain species. So we've had a pangenome for wheat for some time now, and we've brought in the Magic 16 rice genomes as well. So those are already in. And soon we're going to be adding the barley pangenome and the oat pangenome. They'll be coming in the next two releases. So we're starting to see more pangenomes represented. The brassica community has a lot of different genomes going on and are interested in bringing them in. So it's slightly broader than a pangenome, but we're doing our best to kind of bring in more brassica species as we go as well. And I've said here we have them for major crops. That's kind of where we are at the moment. But we'd love to have orphan crops in too. So that don't be put off. And the way that we represent the pangenomes, we're not yet in a graph genome representation of them, but we're keeping a close eye on that space, is we represent them, well, in plants as cultivars, in the vertebrate space as breeds, strains. We have for mice as well, so depending on, so we'll call them cultivars here because we're talking about plants. And we have all the genomes loaded individually, so you can go in and get as much kind of information as you would about an individual genome, or that we have them kind of hanging from the reference, as it were. So if you come here and you're in the rice reference to Nippon Bari, we'll have a list of cultivars, so you can get in and see all of those other rice genomes that are sort of underneath it. And then that has implications for the comparative genomics that we run. So our pipeline is called Compara, and it does, well, I say pipeline, the various pipelines do a lot of different things. We build gene trees using this kind of consensus of different phylogenetic trees based on the protein sequence alignments. So we generate gene trees and we share those. From those, we also predict what are orthologs and what are paralogs by looking at the kind of duplication and loss coming out of those inferred trees. And we also provide whole genome alignments. So LastC has been used kind of traditionally to build pairwise alignments, but we're building more multiple whole genome alignments now where we're using cactus. And if you're, say, if you're in plants and you're looking at kind of a given at a reference genome, then you will have this kind of broad taxonomic range you'll be comparing across all plants. But where we have those pangenomes in, we also offer the cultivar view where you'll just be looking within species. So if we can see that here, if we have in the menus on the left and we've got our plant comparer, we can show in the first menu where we have kind of gene trees and things, it will be that very broad across all of plants. Sorry. But we also have where we have a cultivar view, we can show a gene tree. And so here, this is what we're showing. So we've got all of those proteins have been kind of merged into a single tree. We have an indication here of the conservation of the structure of the gene. And you can see we've got all these different wheat varieties kind of grouped here. And this is just like a very small snippet of what's a very large tree, but just to give you a flavor of the kind of displays that we show. We also provide, as I mentioned, pairwise whole genome alignments. So this is a comparison of the wheat reference against barley. So we can see here we've got the wheat genes, and we've actually got them because wheat is a hexaploid. So we actually have each of the subgenomes kind of showing up for comparison, compared against what we have for the barley reference. So you can start to explore that kind of conservation. So here we're just zoomed in on a gene, but we have it is whole genome alignment. And then with the cactus, we will actually show some what can get quite large alignments. So here we've got one with 16 wheat cactuses. So we've got cactus and some cactus. We've got wheat and some close relatives aligned using progressive cactus. And so again, we can start to see these views where you can see which genes are present or not and how the structures kind of vary across those. So if you're interested in looking at the conservation, you can sort of get in here. And but again, it's whole genome, so you can scroll along and kind of see how that's, you can, well, look at all different locations as you wish. I've just centered on a gene here. So whenever you're in Ensembl, we have a tools menu at the top, or we have a menu at the top where you can get to different parts, and one of the parts you can always get to is tools. And so here we have a list of the things that we offer. So we have searching with kind of hammer or blast, and the variant effect predictor, which is our most popular tool. And what that does from the name, it predicts the consequences of genetic variants. We run it across all of the data that we've imported, but we also have opportunities for users to supply their own variants and run kind of to see what the predicted effects are of those against their genome of choice. And so we have a web interface if you have kind of small number of variants. We also have a command line tool if you have larger volumes of data, and it also is a little bit more extensive in the things that you can do. And with our REST API, we can also kind of support simple URL-based queries as well. And if you have run a VEP job, you'll get out a kind of a list of your variants, the genes in which they're in. So this is actually 2 genes, and then the effects in all the different transcripts that may be within those genes, and a prediction of whether it's something kind of more serious or potentially not having an impact, if it's in an intron, these kind of things. So you can then start to filter on the consequence type. And there are other columns of kind of severity and scoring that you can filter down, so you can reduce down to a set of your most interesting variants if you've uploaded a large set. And then finally, we make everything available for download. So we have our FTP site where we'll have our different releases and different data types and different species. So, we know that we have other, we have kind of users that will come and just take everything after a release and update and kind of build other tools on top of that, which is great. And then for our kind of in-between users, so when you want to do a query that's a bit too big for doing manually on the website, but is not, you're not ready to get into the API or the full download, then we have our Biomart tool. So this allows you to kind of come and choose a genome of interest, choose either a kind of what we call is our filters, but this would be picking maybe a region that you care about or a type of a gene description, a gene ontology term, to filter down to a kind of small set of genes, like a couple of hundred sort of scale. And then you can also decide how you want the output to look. So it will print you out a nice kind of tabular output that you can take away into Excel or whatever else you want to do with it downstream, kind of as you want it. So you can customize it. And this is quite a popular tool that we have across the different component sites. So the other thing that we do is that we partner with other resources. Some of whom are here in the audience today. So Grameen is a platform that we work with very closely. Grameen actually hosts a mirror of Ensembl plants, which we kind of synchronize at least once a year. And then Grameen also has much more extended functionality and including their pangenome sites for grape and maize and rice and sorghum. But a lot of that is underpinned by Ensembl code and obviously the data where it's mirroring our plants directly. Until relatively recently, we've been part of the ViewPath-DB consortium. So we were running all of their data processing for their species, their kind of data load. So we loaded all the genomes and annotations and expression data for all of the species. And then ViewPath was kind of much more on the sort of downstream analysis and community outreach side. With the recent changes in funding, we're not kind of playing an active part in that currently, but we'll see what the future holds. And the other collaboration that we have is Worm-Based Parasite, where they're actually hosted on Ensembl infrastructure. And this is where we have kind of 200 helminth genomes. So it's nice to see that our code is used kind of more widely to underpin some other resources as well outside of Ensembl. And then the Ensembl, as we know it, as you kind of may have seen from the screenshots, is somewhat of an aging code base. It's a project that's been around for nearly 25 years now, and not saying that all the code's been around for 25 years, but it's definitely been evolving over time and is due for an upgrade. So the team have been working very hard to produce the kind of next version of Ensembl. So it's currently in beta. And this is what it looks like if you go to beta.ensembl.org. And the first thing you'll probably notice is that we have a range of organisms here. So we're no longer separating vertebrates from plants, from fungi. We're going to have like everything in together. So this will eventually replace all of those component sites, and this will be the new Ensembl. It's sort of app-based. So the three buttons that you had on that page were species selector, genome browser, and entity viewer. The first time you go in, you will need to select a species to work with. And that's where you can kind of choose from all the species that we have and create and manage a list. And then that list will be remembered when you go back in future visits. So you can keep working with the same species. We have the genome browser is kind of the heart of what we do. So we have a new genome browser there, and what we're calling the entity viewer, which allows you to kind of focus in on one of the genetic features and get more information about them sort of in depth. So when I talked about the species that we covered before, at the moment, because we're in this transition phase between, I mentioned that we've frozen rapid, and we're moving towards this unified site. So our data is sometimes it's in two places, sometimes it's in one place. And so this is a kind of an overview because we know it's a little bit confusing at the moment of necessarily where to find things, and that will improve with time. But this sort of tries to capture where we have our different data right now. So the wedges are the component sites. So we have the vertebrates, we have the metazoa and the plants and the protists and the fungi. And then this blue curve is the new beta site. So you can see from this that we have an overlap. So a lot of vertebrates are in both platforms, but there is an awful lot that's now in beta only, and particularly for metazoa as well. A lot of these are coming from the biodiversity projects that were going into rapid, and now have just rapid's been frozen and they're now in beta. And what we'll start to see is that more kind of new releases will be going straight into beta and data will be added there. If your species is in a component site, I would still recommend that you stay there for the time being because it has more functionality. We still have this kind of things are a bit lightweight still in beta, but over time we'll add more data, add more functionality, and eventually the beta site will become the live site and that will be the place to go. But for now, if you go to a component site and you can't find your species, it's well worth checking if it's in beta because we'll have something there. So when you go into the beta site, we have as I mentioned, you kind of need to start by selecting a species to get cracking. We show our 42 most popular species with icons. Here I've selected breadwheat, which is why that's shown in a different color. So that will kind of stay with us as we go around the other bits of the site. If you can't recognize the icons, you can't find what you're looking for, don't worry. We have a search box and you can add them by text as well. In the top left-hand corner, we have this kind of navigation bar always. So we have the species selector, and whichever part you're in will be highlighted in black. So then we can see how you get to the genome browser, the entity viewer, and the tools we have available at the moment are BLAST and the Ensembl web. And we always have a help icon. And then the new browser is much less colorful than the old browser and much faster. So the zooming capability is much smoother than it was before. Here we can show, again, we're in breadwheat because it's highlighted in black, but if we toggle over to the Japanese rice, we would get the view of the genome browser for that one. On the side, you always have the tray where the drawer, sorry, where you can access additional information and you can move across to the variation. And we have the different strands kind of shown here. If we go more into the entity viewer, we can learn about the function. We can get more to the different domains that it has. We have links out to Pro narrative right now, proteins of a set of species. So it's not compared against everything yet. It's compared against a kind of selected set of species. But this is where you can find that information. And then on all of the pages at the moment, we have a little kind of a smiley face feedback button. So we would love it if you want to go and have a look around, tell us what you think. Do keep in mind that not all functionality and not all data is there yet, but if there's something you desperately think is a priority, we'd love to hear that too. So you can go there and leave us some feedback. And then we have, if you're really interested and want to kind of keep up with us and learn more, we have mailing lists. We have the developers mailing list and the announce. You can reach us at our help desk. And then we have a lot of training materials that are available online, either through the EBI's training site or through Ensembl's training site. So you can follow along with some of that. We keep all our materials live and exercises and everything for all the trainings we get. So that's everything that I wanted to kind of say. This is our whole department. This isn't only Ensembl, but Ensemble's a really big chunk of these people. And we're mostly funded by Wellcome and EMBL, but with contributions from all these other funders as well. Thank you.
Thank you, Sarah. Very great presentation. I think we have a question from Sushma in the chat. Do you have any plan for improving gene functional notation and linking to literature more efficiently?
Well, this is that would be great, right? All of the so much good stuff in the literature and how to connect it. So I think this is something that we've traditionally, because we've been very high throughput and we've sort of not done, I suppose, it's been where we can kind of run high throughput analyses rather than doing curation, but where we can link out to other sources, then that's great. I know some of the other teams at Europe PMC are working hard on the literature mining and kind of AI tools to do this in new and better ways. So hopefully that will provide routes that we can bring more things in. Obviously, Reactome being there and being a great resource as well that we're connected to, and Plant Reactome is I think is where you're coming from, Sushma, right?
Yeah. But still we are more heavily on the manual curation, and I know it is inefficient, so we have to also think about speeding up because it's not sustainable, the manual curation, and funding is also not there. So thank you for the great seminar.
Thanks.
I think Leonor raised the hand.
Yeah, thanks a lot for the great seminar. And I totally can appreciate the overhauling a 25-year-old resource as we've just been doing that with Dyer. I did have a question about the how one sort of gets the new genome in. So I'm thinking specifically about this new genome annotation for Arabidopsis, which we're, I think, very close to finally, getting that into GenBank. But sort of like, what is the procedure for sort of keeping these things in sync? So that, because I'm concerned, I think, about making sure that like the reference genome is the same for all of the places that represent it. So do we like write to you and say, hey, the new genome annotation is in?
Yes, that's a really good way. Direct communication we love, because then also when we have questions, we have a person connected to it. So that's a really good option. I think something that we're starting to do, well, so first we can only, in terms of the assembly, because it has to be submitted into the archives. That's one of the reasons, so that everybody's working on the same assembly and you know that you are. So that's kind of the where that originally came from in the early days of the human genome, when there were various versions. That just kind of simplified everything. So there's part of that. But for the annotation, we don't necessarily have that, because the annotations, many people do not deposit their annotations when they deposit their assembly. So you can't link back to them. So because we get them from various sources, that is, we prefer it if we can link back to a deposited annotation and say that's where it is, and we would love that. I think we're doing kind of more of a push in plants. It's been more, it's kind of, I suppose, less high throughput in the way that we've brought things in, and more interacting with the community, sort of where people have reached out to us or we've reached out to them and said, we've read your paper and we want to bring it in. But we're sort of moving towards more high throughput, kind of deciding, like finding what's out there, and then we'll evaluate it, rather than kind of the other way around. So hopefully we will pick up on things sooner when they have been deposited, and then be able to be more proactive in reaching out to people. That's where we're sort of moving. But yeah, obviously we're well aware that Arabidopsis is coming, and I think what we will probably do in that case is we would definitely want to bring in the new one, but we probably wouldn't retire the old one immediately. When we have a genome that's been really heavily used, so we've done it for wheat as well, we've still actually maintained the version that is our most popular and brought a second one in. So kind of breaking our, like, where we've always had one, because we don't want to leave people without the thing they're used to. So we'll kind of maintain both for a time, and then at some point switch over, which is the like one that we steer people towards.
Thanks, Sarah. Question in the chat. So Dana is asking, what will you be calling the new genome browser tool?
Well, it will be Ensembl. I mean, at some point, current Ensembl will become old Ensembl. And it won't be confusing at all. It's going to be fine.
Okay. Then Eti is asking, could you say more about the pangenome data? Are these calculated by various group and submitted? Are these genome level pangenome or pangene analysis? How does one see them?
Okay. So we have a little bit of a mix going on, truth be told. So my colleague who's looking after the sort of human pangenome, that pangenome has come in from the community. It is the Human Pangenome Reference Consortium. It's their pangenome, and that's sort of protected, and that is what it is. For us with wheat, we've taken the choice that we've brought in the 10 plus genomes. We've brought in the kind of UK contributions to that project. We've brought in a few wheat genomes since, and we're like adding them in. So we're kind of, but because we're not doing a pangenome, we're not doing a graph representation, we're not doing a kind of explicit set. What we're doing is comparative genomics between all of those members. So we sort of like can do that without changing anything, the relationships between the pangenome, by like adding more in. We're not actually changing anything. So that's how we've done it so far. Sorry, with wheat. What we're doing with rice, which I had a slide but I didn't show, was we have a project, the Panorizer project, where we are taking the Rice Magic 16 and making pangene identifiers for those genes which are kind of we are clustered together using the method that we've used that we've published, kind of related to that project. And then we've got a pangene identifier, which is closely based, but not entirely the same as the recommendations that came from the Ag Biodata group. So we took a lot of advice, but it is slightly different in terms of how we're using the species identifier. But the rest is very similar to the recommendations from the pangenome group, which was very helpful to us. And then those are going to be shown as synonyms attached to the genes, so that then you can search for a pangene identifier and get all the genes back from it, or search for a gene and get back kind of which pangene it belongs to. And in the pangene identifiers, we have like the species, we have the group that generated it, we have a version, and then we have your kind of identifier. So we're trying to capture all that information so that when there are multiple different pangene sets that have been generated from different sets of sequences, we can differentiate those, or even if it's the same initial genomes but a different group with a different method, we can differentiate that. So that's what we've kind of tried to do. And then the way that you can see them, so that's how you would kind of see them from the identifier level. So the rice is not online yet. It's coming really, really soon with the paper that's also we're working on furiously. But otherwise, then yes, it's through the comparative trees and the cultivar views is where you can have a look at the kind of gene level and see the similarities, because that's how we're representing it. If I captured everything there, sorry, Ethy, there was a few bits.
Yeah, that's good. Thank you. I will likely have more questions as time goes on.
Nice, ping me. That's good.
Okay, will do.
So thank you, Sarah. So Andrew asked, when will the beta site be made available for hosting data managed by other groups, Gramine or sorghum base?
Well, all the code is already in GitHub if you want to kind of get in there, but I wouldn't recommend it just yet because it's still highly actively being developed and functionalities being added on. So I would sort of hang fire yet and wait for things to settle a bit more. That would be my kind of recommendation for now. But yeah, I mean the code is available and it will continue to be there. So once it gets to a point internally, we're sort of looking at that tipping point where we will stop, say for plants, adding genomes into Ensembl plants and begin only putting them into beta. We're not there yet because we haven't got all the functionality that we want. But at the point that we are, that's maybe a good point for the adoption to happen. But obviously, Grameen, we're going to be speaking with you closely, and I think we have plans to catch up with you. I was asking this internally, when is a good time to do it, and we will be reaching out to you in the new year.
Thanks.
So thank you, Sarah. Then Sebastian says, thanks, Sarah, for the updates. Looking forward to comparing against many more pangenomes. Are you looking to include more of those or focusing on crops at the moment? And additionally, what in your view makes a good quality pangenome?
So far we have the ones that we have. We have the kind of 4 that we're on, and we're in contact with the community to see if they're ready to bring in, and these are sort of quite like well-established pangenomes, I suppose. We are by no means limited to just crops. We have not exactly a pangenome, but we've got a Drosophilidae level for flies kind of going on in Metazoa as well. And we would be happy to bring in other things. What we have battled with in the past was the scale of bringing in. So for instance, if we've got 70 genomes coming in for the same species, that's been a lot for the team to import all at once. But we have now kind of overhauled the system, and that's actually not such a big job anymore. So that will make it easier for us to bring things in so we can be more welcoming for different pangenomes, because we're in a better position now with our setup. So yeah, if you have pangenomes that you're interested in working on, we'd love to hear from you, and we can decide. In terms of what makes a good quality pangenome, so we're still thinking about pangenomes where each of the reference is kind of a good quality. We haven't brought in the kind of, where you have a good quality reference and then you have bits and pieces coming from the sort of like missing parts coming from other genomes, the sort of more like the original way that groups were generating pangenomes. So you'd have one reference and plus extra parts. We haven't brought in any of those. And I think many more pangenomes that have been generated where all the individuals are kind of, they have the haplotypes and we have the genomes. So whether we would go back to any of those, I'm not sure. But for now, we're kind of focusing on the ones where the individuals are high quality. But as I said, with the wheat, what we're doing, where we're bringing in what's available, doing it through a comparative genomics way, and we can just keep adding things on, then that's also something. It doesn't have to be like a very comprehensive, even pangenome. If there's benefit to bringing it in and it will kind of grow, that's also something we can think about. Although recalculating the comparative every time we add something new is a cost that we have to consider. So we might not update every release, but it might be something that we do kind of periodically.
And then I think there's a suggestion from Dyer. One humble suggestion for the beta ensemble page: the feedback symbol looks too much like the Amazon logo and doesn't quite convey the idea that it is a button to provide feedback about the website.
Thank you for your feedback on our feedback. I will pass that on.
Are there any other questions for Sarah? If not, I'm going to thank Sarah for the great seminar and for concluding in an excellent way this webinar series of 2024. And I wish you all a great day and to see you at PAG if you will be there. And if you have any question about Ensembl, please feel free to reach out to Sarah. And thank you so much for joining.
Thank you. Bye.